PCR and gel electrophoresis are very important tools in molecular biology. Today, we will perform beta-actin specific PCR amplification from complementary DNA and downstream agarose gel electrophoresis. Beta-actin is an important housekeeping gene. Before PCR, we will mix all the PCR raw materials to make PCR cocktail in laminar airflow system to avoid any contamination. We mix all the necessary chemicals like PCR master mix, forward primer, reverse primer, nucleus free water and complementary DNA in specific amount to make the final volume 10 microliter or 20 microliter. If we don't have PCR master mix, then we have to make it manually by mixing Taq DNA polymerase, Taq buffer, DNTP mix, magnesium chloride and nucleus free water in specific amount. All the mixing is performed in minicooler and reverse pipetting should be done each time. This is DNTP mix. DNA polymerase, Taq DNA polymerase. This is beta-actin primer, forward primer. Tubes are ready for PCR. Just before inserting the tubes inside PCR hood, tubes are spinned a little bit. This is the PCR machine manufactured by Applied Biosystems. We just turned on the machine. Clicked on run by pressing F1. Then the display shows the saved protocols of previously run methods. Beta-actin specific PCR is also saved here. We will just click on view to check the temperature points and the time intervals. We are now keeping the samples inside the PCR hood. We will start now. Final volume is 10 microliter. Now the cover is heating. After the heating is completed, the PCR will start automatically. PCR has already started. The steps are initial denaturation 95 degree, then final denaturation, then beta-actin primer annealing temperature which is 48 degree, and then initial extension 72 degree, and 95 degree, 48 degree and 72 degree. These cycles will be repeated for 40 times, that is 40 cycles, and 72 degree is the final extension and 4 degree hold. For agarose gel electrophoresis, we first make agarose gel. To make the gel, we make a weight by volume solution by weighing agarose and then dissolving this in 1x TAE buffer by heating. We first measure 0.375 gram agarose in 25 ml of 1x TAE buffer to make 1.5% agarose gel. We actually make and solidify the gel before the completion of PCR because it takes time to solidify, and we also have to give a pre-run before the sample loading and final electrophoresis. We are now weighing agarose. Weighing of agarose is completed. This is 1x TAE buffer. We will measure 25 ml of TAE buffer in a measuring cylinder and then pour it inside a conical flask and then add agarose. This will make a whitish solution, but after heating in microwave a clear solution will be made, that is agarose is dissolved in buffer. Ethidium bromide is a good DNA intercalating agent that we make use widely. We will mix it in gel and pour. Look, PCR is in final extension step and we are waiting for the fall of temperature up to 4 degrees centigrade. After that we can store the samples in 4 degrees centigrade. Gel has solidified and we will place it inside the gel apparatus. Now we are going to give a pre-run which will be accomplished at 100 volt for 10 minutes. Before that, we are now preparing the gel. Then it will be placed inside the gel apparatus. We are now pouring 1x TAE buffer as running buffer and then placing the gel. Watch carefully that the wells are facing the cathode end. Black wire is connected to cathode, which is negative electrode, and red wire is connected to anode, which is positive electrode. PCR has completed. We will now take the samples out and keep in mini cooler. This is 50 bp DNA ladder. Free run is being continued. The bubble formation is indicating that free run at 100 volt. We frequently use glycerol to mix with the samples and load into the wells. Ten minutes of free run has completed. We are now turning off the current. Now we will load the samples mixing with glycerol. This is 50 bp DNA ladder. First we will load the DNA ladder into the leftmost well. Loading should be worked out carefully, as the microtips can damage the gel. Now we are mixing the samples with glycerol. It will help us to pour the samples inside the well very easily. The samples are being loaded into the well. Then again we turn on the current, setting voltage to 65 volt. Now gel electrophoresis has completed. We are now taking the gel out of the gel apparatus for imaging in gel dock. This is the gel dock machine connected to computer. We will place the gel on the UV tray. We generally keep the wells in the upside position so that the wells enters first into the tray chamber. While placing the gel on the UV tray, we must avoid bubble formation. Then we run the software after inserting the tray inside Image Lab software. Then we will click New Protocol. Then select nucleic acid gels with ethidium bromide. After imaging, we will get an image like this. This is the typical band of beta actin, which was the PCR product.